we have now reached the, as some colleagues call it, age of microfluidics, where you can test different conditions, different setups on organ platforms. And there's currently sort of 3 major organ platforms available. One is called precision cut tissue slices, where you actually take the tissue of the host and have slices somewhere in the range of 100 to 400 micrometers.
Welcome to the Swine Health Blackbelt Podcast, the latest swine health research digested for you. My name's Dr. Clayton Johnson, and I'm the host of the podcast. Joining me in this week's episode is Dr. Dirk Werling. Dr. Werling is a professor of molecular immunology and the director of the Center for Vaccinology and Regenerative Medicine at the Royal Veterinary College at the University of London. Dr. Dirk, thank you very much for joining us. Why don't you start with a little introduction for the audience?
Yeah, will do, Clayton. Thank you very much for the invite and giving me the chance to talk a bit about our latest development. But my background is fairly straightforward, actually. So I studied veterinary medicine in Hanover, in Germany, then moved with my job the world a bit. So I worked in the US, in Africa, in Switzerland, and now since 2003, I'm in the UK. And my group is really investigating how we can improve vaccines in farmed animals and how we can make sure that we really have the best vaccine composition. What I mean by this is I have no doubt that we have really good vaccine antigens at the moment, but I think that they are potentially not fit for 2050. Yeah, I think, and what I mean by this is if you take into account what mankind has achieved in the last couple of years due to the pandemic and how quickly we showed that vaccine can come on the market and how quickly we can incorporate new technologies into vaccines, I think that there will be a quantum leap in veterinary medicine happening within the next couple of years forward. So this is what we're working on.
A full value relationship starts with understanding your business, and Elanco knows growing the healthiest pig requires focus on every segment of production. Through continuous innovation, trusted solutions, and actionable insights, Elanco is invested in helping you achieve the full value of every decision. Their portfolio offers solutions that manage disease challenges, minimize variation, and mitigate mortality to optimize pig health. Get full value from start to finish with Elanco.
Well, and I would think, Dirk, that vaccines are only going to become more important with time. With the pressure that we have on the use of antibiotics to control disease, we're going to need really robust vaccine platforms in the future. And that's maybe what you're suggesting, that what we have today got us here, but it may not get us there to what our future needs are.
No, I completely agree with you on that statement. And I think this is exactly, we may have become, let's call it a little bit lazy in terms of that, because in terms of developing new vaccines, because we had the antibiotics as a backup, or actually we had the antibiotics as medium to prevent disease outbreaks in the prophylactic or metaphylactic treatment scenario. And I'm not saying that antibiotics don't have the purpose. If an animal is ill, by all means, it needs treatment. But I think that if we combine our knowledge about the microbiome we have, plus our knowledge with a new vaccine, then actually, and it may sound a bit arrogant with that, but I would postulate there is no need for prophylactic or metaphylactically used antibiotics anymore, which in theory, you could argue that is the best case scenario we can have because it keeps our pigs more healthy from the beginning on. We can produce products where the consumer is more reassured that this is coming from healthy pigs without the potential residuals of antimicrobials in there. And I think what is really important is we also will reduce the number of antimicrobial resistant strains which are coming out of not only farmed animals, out of human medicine as well, which have an impact on the environment as well as on the bacteria in the environment.
I think everybody's bought into that 100%, Dirk. So what are you guys working on? What are these novel approaches that we can look forward to?
Yeah, that is a really cool approach we're using, and we're actually picking up from human medicine. So one of the big issues we have always is testing vaccines in large enough animal numbers. So if you develop a vaccine, you have 3, 4, 5, 6 different compositions, and you need to have enough animals to actually figure out which potentially works best. We have now reached the, as some colleagues call it, age of microfluidics, where you can test different conditions, different setups on organ platforms. And there's currently sort of 3 major organ platforms available. One is called precision cut tissue slices, where you actually take the tissue of the host and have slices somewhere in the range of 100 to 400 micrometers. And that means you have the tissue in its original 3D composition. You can have organ on a chip platforms, the real microfluidics, where you have cells of your host, you put them in a little chip which is not bigger than this, with a total volume of around 100 microliters. And the advantage there is that you can start with a defined cell population, and you can have the cells in a flow, and you can actually add shear stress and things like this into the whole system. And then you have the last platform, the so-called organoids, where you recreate a specific organ under the impact of growth hormones or growth modulators in vitro completely. And all these 3 platforms allow you to test different approaches on the target tissue from the target host under very defined conditions, which means that you can actually pick and choose what is the potentially best candidate to take forward into the in vivo testing phase. And that has far-reaching effects, because not only do we have a lot more basic understanding on how the vaccine interacts with tissue, how the pathogen interacts with the tissue, how the pathogen interacts with tissue from vaccinated animals, but it also means that we can reduce the numbers of animals necessary for the in vivo testing. So we're picking all the 3R components, and we can also reduce the number of mice or rodents, which are not the most ideal species for pigs to test vaccines in. And we can really speed up the whole process with hard facts on what is the best candidate to take forward. And I think that is really exciting times, because we're not only pushing the boundary of our basic science understanding, we really can transfer that a lot quicker into the real world.
That sounds super exciting, Dirk. If you're using tissue, so cellular-based tissue from individual animals, I would presume you can actually have treatments and control within the same animal in that process, right? So you're not comparing different animals in different groups. It's truly the same animal cells that you're looking at as the treatment and controls.
Exactly. And that's exactly where I'm getting excited about as well. So just to give you an example, we are running currently trials with precision slices, where we generate slices from lymph nodes, from the lung, from the same animal. And we can have treated slices and untreated slices from the same animal. So we can already have outcomes from 2 different organs from the same animal, and can assess what happens in these different tissues. Now, if you then add in all these multi-omic platforms we have afterwards, you can really pinpoint whether you get reactions in all the tissues which are the same, and which you can potentially pick up in the periphery, which means you can not only develop the vaccine, but you can also develop automatically the pen site testing device and see whether the vaccine has actually worked. And that allows you then subsequently as well to discriminate vaccinated versus naturally infected animals, which I think is really important if you think about the big threats such as African swine fever, FMD, everything where pig producers go, whoa.
Yeah. Diva vaccines are part of the holy grail of vaccine technology, and even beyond the foreign animal diseases. You mentioned PRRS, right?
Yeah.
Being able to vaccinate breeding stock for things we don't today because the testing becomes problematic, that would have tremendous value.
Yeah. And I think this is really where these organ platforms will be extremely handy. If we come with subunit vaccines, we need to understand how they work. We need to understand how we can adjuvant them potentially to have a real protective and functional immune response. But then also, if you link this with the development straight away with the pen-side testing device, you can offer the producers the whole set by saying, okay, we have the vaccine here, you can test with that. And if you then link in all the new technologies like sound recognition and so coming, I'm guaranteeing you that the producer can save a lot of money on treatments.
Very good.
Salmonella presents significant challenges to pig health and performance and poses food safety risks to humans. As the first and only vaccine offering live attenuated strains of both Salmonella cholerae and Typhimurium, Enterosol Salmonella TC from Boehringer Ingelheim protects pigs against both serotypes with a single oral dose. Talk to your Boehringer Ingelheim representative to learn more.
Dirk, as you evolve this technology, do you envision that your group or others will do this as a service for existing vaccine technologies, or do you think that this is going to spawn kind of a new generation of vaccine producers?
I think it's going to be a bit of both, because as I said, we have good vaccine antigens at the moment. But what I sometimes wonder is whether these vaccine antigens are in the best composition.
Yep, delivered the most optimal way.
Exactly. So I think revisiting some of the, let's call them for the sake of it, old vaccines in a new composition, this is something we can test on these organ platforms very easily. But also when you think about what we have achieved with the mRNA vaccines against SARS-CoV-2, now if you want to have something like this for ASF, FMD, we need to understand what is the best composition, what's the best way of long-term protection, what's the onset of immunity, the duration of immunity. And I think that the organ platforms will help answer some of these questions relatively easy compared to in vivo models. So I think these models really will have the right to exist in adapting old vaccines or adjusting old vaccines, as well as being used for the development of new vaccines.
Tremendous. It's awesome work you guys are doing, Dirk, and I can't wait to hear more about it as you guys make progress.
I would be more than happy to update you on that in our next podcast.
I look forward to it. Thank you so much, Dirk, for coming on to our show, and to the audience out there. Thank you for listening and making what we do possible here at the Swine Health Blackbelt Podcast. Please check out our website at swinehealthblackbelt.com. And don't forget to subscribe to the podcast so that you don't miss out on any of our great episodes coming to you every week. For Dr. Dirk Werling, I'm Dr. Clayton Johnson. Thank you very much for joining us. And Dirk, I think it'd be most appropriate if we end this with cheers.
Yay, up for the new technologies. Save the pig. Bye.
Bye-bye.
Hey everybody, we're always searching for the latest and greatest research to share each week. If you have a swine health related research trial and would like to come on the show and talk about it, share it with us, please feel free to email the research to hello@wisenetics.com. That's HELLO@WISENETIX.com.